Good morning. My name is Divya Pradhan, and we are going to talk about the pancreas. The pancreas is a soft, tongue-shaped gland that extends across the abdomen, and the position of the pancreas is retroperitoneal, and it lies towards the greater curvature of the stomach. The most interesting aspect about the pancreas is that it is an endocrine as well as an exocrine gland. Of course, we're interested in the exocrine function because we're talking about digestion. The exocrine function is that it secretes pancreatic juice. The pancreatic juice is produced by a special group of cells called acinar cells that lie in clusters around small tubes. These tubes drain into larger ducts that join together to form one single pancreatic duct that drains the juice into the duodenum of the small intestine. The connection with the small intestine is usually at the same location where bile duct connects. This is important. You'll understand why later. And the connection, they join together to form a small raised structure called the hepato-pancreatic ampulla. This is a spelling mistake. This should have been spelled as hepato. Pancreatic. This is a C. Ampulla. And this opening of the ampulla is guarded by a muscle, and that is called a hepato-pancreatic sphincter. As you can see from the picture, in the pancreas, there are these acinar cells that are draining into the small ducts, which I said join to form the larger pancreatic duct. Now let us talk about the composition of the pancreatic juice. About 1200 to 1500 mils, that's a lot, of pancreatic juice is produced daily. It consists of water, enzymes, and ions. The epithelial lining of the pancreas produces bicarbonate ions, and what that does is it makes the pancreatic juice alkaline. In other words, it raises the pH. Normally, the amount of hydrochloric acid produced by the stomach is exactly balanced by the amount of bicarbonate ions secreted by the pancreas. The bicarbonate ions has three functions. It neutralizes the acidic chyme that's coming from the stomach. It provides the optimal pH for the intestinal enzymes. And next is it blocks the activity of the pepsin, if you remember, that's coming from the stomach, and thus prevents the pepsin from digesting the walls of the duodenum. Now let's look at the enzymes. The pancreatic juice contains enzymes that digest carbohydrates, fats, proteins, and nucleic acids. The first is the pancreatic amylases, which are exactly similar to the ones that are found in the mouth, produced in the saliva. Function is the same. They digest carbohydrates, more specifically starch or glycogen is broken down into disaccharides. The lipases break down fats into fatty acid and glycerol. Here, if you remember, in the stomach also you had a little bit of lipase production, but the digestion of fat was poor. Why? Because fat, before it is digested, it has to be broken down into smaller pieces. So large lump of fat is broken down into smaller pieces, and this is called emulsification of fat. And this emulsification is done by the bile salts. break down a big piece of fat into smaller pieces so that there's increased surface area for the enzymes to act on it. Remember, fat is not soluble in water. Therefore, this step is necessary. The protein digesting enzymes are called proteolytic enzymes, or also called proteases. Are three of them: trypsin, chymotrypsin, and carboxypeptidase. Each cleave the protein chain at a particular site. So there's a big chain of amino acids, and each of these cleave at a particular site so that individual amino acids are formed. The most interesting fact is that these are stored in the acinar cells as in inactive forms. These are called zymosin granules, and the zymosin granules are activated once they reach the intestine. So the trypsin, for example, is secreted as trypsinogen, and this trypsinogen is activated, becomes trypsin, by the action of enterokinase, which is produced in the small intestine. Enterokinase is also called enteropeptidase. Once trypsin is formed, trypsin will activate more and more trypsinogen. That means it will cause more and more trypsinogen to become trypsin. It will also cause the zymosin granules, which store chymotrypsin and carboxypeptidase, to form more and more chymotrypsin and carboxypeptidase. The last enzyme is nucleases, which breaks down nucleic acids into nucleotides. Now the regulation of pancreatic secretion. The regulation is under two controls, nervous as well as hormonal. So during the cephalic and the gastric phases, the nervous system is active. In other words, parasympathetic nerve impulses stimulate pancreas to secrete more and more digestive enzymes, and therefore more and more pancreatic juices. As for hormonal control, there are two enteric hormones, secretin and cholecystokinin. Secretin is a peptide hormone which is secreted by the cells in the intestinal mucosa when acidic chyme enters the duodenum. This triggers release of bicarbonate ions from the pancreas to neutralize the acidity of the chyme. Very important. Cholecystokinin is the second peptide hormone produced in the duodenum in response to protein and fats. It stimulates the release of enzymes. It will also stimulate the gallbladder to squeeze out more and more of the bile salts, so that more bile juice pours into the duodenum. Lipid digestion therefore can take place. It also relaxes the hepatopancreatic sphincter. If you remember, we talked about it, so that bile and pancreatic juice can now enter the duodenum. When no digestion is taking place, that sphincter, the hepatopancreatic sphincter, remains closed. There is no need for that digestion to take place. I thank you for listening to me, and with this we come to the end of the digestion by pancreas. And you have a great day.